I am Dakota Parnell,
And I am Jesse Orell.
and this is our joint project on constructing schematics for a novel microfluidic slide for in vivo imaging of larval zebrafish.
Individuals chronically consume high-fat diets. It is now becoming clear that early dietary lipid uptake can affect us throughout life. Shorter life expectancies and type 2 diabetes are examples. Study is in critical need to understand how high-fat diets affect intestinal organelles.
Zebrafish have previously been studied for a diet-induced obesity model. However, research of single-cell resolution is lacking. Chronic high-fat diet-induced obesity in zebrafish shares common physiological pathways with mammalian obesity. Lipid processing within the larvae's intestine impacts the whole animal metabolism. Lipids are absorbed by enterocytes where they are sorted and packaged en route to the blood. How cholesterol is taken into enterocytes and packaged in lipid droplets is unknown.
Here's a close-up of a larvae intestinal tract. This image was taken with a transelectron microscope, or TEM. The lipid droplet shows that the larvae has been fed. The lipid droplet is absorbed through the apical cells of the brush border from the lumen into the enterocyte. The lipid droplet is absorbed through the basal membrane at the bottom of the intestine and ultimately reaches the bloodstream. The metabolism of the lipid droplet is the main focus of our research.
Zebrafish as a model organism. They make good models because they are optically transparent in larval stages up to 14 days post-fertilization. They reproduce quickly and in large quantities. They share 70% of genes with humans, and of the 70%, 84% of the genes shared are known to cause disease. This 84% is significant for our research of diseases and disorders of lipid metabolism.
Previous methods to image zebrafish were limited to embryogenesis and early larval stages. Our goal is to design a novel microfluidic slide for mounting zebrafish that greatly extends the duration of time for in vivo imaging of zebrafish larvae that are 6 to 7 days post-fertilization, while supplying drugs or nourishment that are fluorescently labeled to observe unicellular processes. The ideal mounting slide will maintain the larvae in a mostly static condition, have channels to provide media exchange, nourishment, and/or drug treatments, and channels to flush waste and any lingering test conditions. The Walters Lab has developed methods to track key lipid processing proteins and dietary lipids during absorption.
3D chamber design. Initially, we used a CAD, computer-aided design, to design what we envisioned our slide should look like. The channel on the left, leading to the head, is the entry channel for fluids, indicated by the red arrow. It is important that the fluid exit channel, leading from the tail, must be past the anus for proper disposal of waste and fluid, indicated by the blue arrow.
Depicted here is a theoretical view of a larvae in a chamber under a microscope mounted laterally. Entry channel coming from the left side to the head, and the exit channel leading diagonally to the right. Here, highlighted in blue, is the intestinal tract of the larvae in a good position for imaging. We can also see, highlighted in purple, the contour of the chamber to accommodate the shape of the larvae.
Chip prototype version 3. Upon testing, we found the larval chamber was too large to properly accommodate the larvae indicated by these red arrows. The excess space in the chamber allows for unwanted movement of the larvae, which could lead to dislodging from the larval chamber. Because of the excess space and allowed movement of the larvae, it would create a blur and we could not obtain clear images of the larvae.
Because our previous prototype was too large, we imaged 10 7DPF larvae using a Zeiss Discovery V8 microscope and the Zeiss Pro software. After imaging, labeling, and measuring of the larvae was conducted, all measurements were recorded and averages were calculated for the 7DPF larvae. Each blue box represents a separately measured dimension. From our calculations, Dolomite printed a new chip, version 4. Dolomite can make a fully functioning, fluidically sealed chamber from glass at such small dimensions. Upon arrival of the new chip, version 4, 7DPF larvae were mounted into the chambers to determine if it was to correct proportions.
7DPF Fish 1 dorsal and lateral view. The top image is the dorsal view of the larvae. The bottom image is the lateral view of the larvae. The pictures shown were used in data collection to determine the new chip dimensions.
This is the most recent version of our chamber that we are in the process of testing. In retrospect, it does not seem like a big change from the previous version, but microscopically it proves to make a big difference. It is almost half the size of the previous version. A 7 DPF larvae was mounted and imaged in the new chip as shown. The chamber shows to fit the fish much more adequately than the previous version, outlined by the blue dashed lines. The red arrow shows little excess space that gives room for waste to escape.
The mitose P pump is used to supply fluids, such as embryo media, and different drugs and diets to the resealable chip interface by connecting tubes. Gasket and pressure of the interface on the chip makes the seals watertight. The diets can be fluorescently labeled for easy view inside the intestine of the larvae. A multi-chamber fluid supply can be running so that up to 3 different diets can be fed concurrently.
This movie is of a larvae in our new chamber, version 4. Liquids are run through the mitos pressure pump into the chamber. It is important to keep the larvae healthy and alive while in the chip. We can see this proof by movement of the blood through vessels and by the heartbeat, shown here. Then we have the brain. The larvae swim bladder. And most importantly, this is the larvae's intestinal tract and the main focus of our research.
This is a close-up movie of the larvae's blood vessels, muscle tissue, and intestinal tract. Note the direction of the blood determining between the arteries and the veins. In the muscle tissue, we can see striations here. And this is the lumen where the food is stored. It contains enterocytes, and the enterocytes digest lipid droplets.
Here are some confocal enterocyte still images. We hope to do this in real time with fluorescently labeled lipids depicted here in green.
Future directions. We hope to be able to run high throughput experiments that can maintain different simultaneous inputs. This will allow us to test a vast array of drugs and diets, which has a vast implication of possibilities to study digestion at single cell resolution for long durations. The example here shows diet A, diet B, diet C, and diet D being fed to 10 fish each with respectively labeled in and output channels.
In conclusion, our project started with imaging and measuring 7 DPF larvae to determine a correct chamber size. After the chip was printed, we mounted larvae into the slide to pump fluids through the larval chamber to the larvae. Our next step is to feed high-fat diets to the larvae that are mounted into the chip and image single-cell processes. We can fluorescently label lipids to track or monitor the unicellular processes shown in the enterocyte here.
We would like to give a special thanks to Dr. James Walters and Gabriella Hubbard for assistance with our research. We would like to thank Ashley Reed and Syed Monas Ali for previous research and background development. We would also like to thank McNair Scholars Program, Bluefield State College, WV Embry, HISTA, and Walters Lab for making this possible. Questions. You may comment in the comment section below, or you may email us at the following address in the presentation. We will provide an answer in a timely fashion.
And thank you for your time.